So good evening, everyone. I'm Lori McMahon. I'm the Dean of the Graduate School. Welcome to this evening's version of Discoveries in the Making. And I think some of you are attending for the first time, and so I'll just give a little background. So Discoveries in the Making is an opportunity for our doctoral students and our postdoctoral fellows to share their discoveries with a non-expert audience. And we feel really passionate about our students being able to learn to speak to a non-expert audience. We think it's our responsibility as individuals who receive federal funding, taxpayers' dollars to help us with our research. It's our duty and our responsibility to share back with a non-expert audience how we're using those dollars, how we're generating new knowledge. And so that's what we do in Discoveries in the Making. And so that's what we're doing. We are making discoveries. We're along the way, along the path to new discoveries. So we've been hosting Discoveries in the Making for about 3 years now. And we started with Discoveries in the Making and local public libraries to talk to a non-expert audience there, and also in a local coffee shop. And about 2 years ago, we transitioned to the Lumbar, which is a science-inspired cafe, or I would say a restaurant bar, to share with the local public there. And moving to the Lumbar was really inspired by Pints of Science. So those of you who are familiar with the UK, a beer is a pint. And so Pints of Science was started in the UK, where a non-expert lay audience would attend a local brew pub and have a beer and listen to researchers, scientists describe their research and the knowledge they were generating. So we cannot be in the Lumbar right now due to COVID, but we cannot wait to take Discoveries in the Making back in person. But doing this through Zoom has allowed us to speak with individuals who are not in town in Birmingham. And so I think when COVID is under control and we feel comfortable being back out in public, we will probably try to do a combination of in-person and Zoom simultaneously so that we can reach people who are across the country and even across the globe. We have had members join us from Antarctica, from Scotland, from New Zealand. So it's not limited to just the local Birmingham area. So I know Luke is excited to share his discoveries in the making with you this evening, and he will enjoy answering all of your questions. This is about a conversation. This is about you asking our researchers, our scientists questions. So he will answer your questions. You can either unmute yourself and speak up, or you can put your question in the chat box, and Dr. Creel and I will monitor the chat box. So the title of Luke Dunaway's presentation this evening is The Discovery of a Salt Sensing Molecule. Okay, Luke, take it away.
Thank you. Yeah, thank you everyone for tuning in on a Tuesday evening to hear about why you shouldn't be eating salt. So like she said, I'm going to talk to you about some of the data we've generated over the last couple of years in the lab. I've kind of broken the talk down because it is kind of hard to interact and jump in with questions to have question points throughout the talk after each of these sections. So I'm going to start off talking about what we knew going into this project, some statistics about how salt affects cardiovascular health and nitric oxide signaling. I'm going to then move into what we learned from this project, what questions we had, and talk to you about a molecule called Hdac and how that's responding to a high salt diet. And then I'll talk to you about what we're working on now. So some unanswered questions and some half-answered questions, the discoveries that are currently in the making. And after each of these sections, I'll kind of have time for people to jump in and have questions. But of course, jump in anytime or just put them in the chat. So eat less salt. You probably have heard this before, whether it's from a doctor hoping that you'll be able to lower your blood pressure, or a concerned family member, or maybe just that health conscious friend on Facebook. You've probably heard that you should be eating less salt. And to put some numbers on this, the American Heart Association recommends that we don't eat any more than 1,500 milligrams of sodium a day, but the average American is eating over twice this, at 3,400 milligrams of sodium a day. So well over the recommended amount. And this, of course, affects our cardiovascular health. And cardiovascular disease is the leading cause of death worldwide. So this is very much a global problem, but it's also a local problem here in the Southeast and in Alabama. So this is looking at cardiovascular disease death rates in the years 2016 to 2018 at the county level. And you can see in Alabama, Mississippi, Louisiana, Arkansas, almost every county is bright red, showing we have some of the highest incidences of cardiovascular disease deaths in the country. So we know salt leads to an increase in cardiovascular disease, but how does this happen? So your first thought may be blood pressure. I think it was my first thought coming into this project in the lab. But not everyone's blood pressure changes with a high salt diet. So people who have high blood pressure when they eat a lot of salt, we term these people salt sensitive, meaning their blood pressure is sensitive to changes in salt in the diet. And if you just think about people who have normal blood pressure, only roughly 1 in 4 of those people are salt sensitive. And if you think of people with high blood pressure, it's a lot higher. It's 2 out of 3. So there's a lot of wisdom in reducing the amount of salt you eat to lower your blood pressure, but this certainly isn't true for everybody. And these people who are salt resistant still have increased risk of cardiovascular disease. So what's happening in these individuals? If it's not blood pressure, what is it? So I want to talk to you about a molecule called nitric oxide. So it's a simple small gas. It's nitrogen and an oxygen bound together. And you actually lose nitric oxide in your blood vessels when you eat a high salt diet. So a quick point of clarification, sometimes when I say this, people think of laughing gas, they're thinking of nitrous oxide, but we're talking about nitric oxide, which is a little different. And so this is a molecule, like I said, important for a healthy cardiovascular system. And it's produced in several parts of your body, but we're gonna talk tonight about your blood vessels, particularly this inner layer of your blood vessels. So your blood vessels have up to 3 layers, the adventitia on the outside, the media in the middle, which is mostly smooth muscle, and then this inner layer, which is the intima or endothelium, as I'll be referring to it tonight. And it's these cells that line the inner layer of your blood vessels, the endothelium, where nitric oxide is really important. So we're gonna zoom in a little further deep into these cells and think about what's happening at a molecular level. And so a quick definition to bring people up to speed is I'm gonna be talking about enzymes a lot. And you can just think of enzymes as these doer molecules. They're these molecular complexes that get stuff done. Here's a couple of drawings of a few enzymes. So you can kind of see they come in all shapes and sizes, and they do all sorts of things throughout the body. So they're in your digestive tract, helping you break down your food. They're in your muscles, building up new muscles when you exercise, but also providing the actual contraction of your muscles to allow you to lift and move things. Some enzymes regulate other enzymes, and they turn other enzymes on and off. And then some produce messages, allowing cells to communicate to one another in the body. So nitric oxide is one of these messenger molecules. And so here we're zooming into the endothelial cell. Let me put my pointer. We're zooming in here to an endothelial cell that's in the inner lining of your blood vessel. And here, nitric oxide is produced by an enzyme called NOS3, NOS3, to signal to other cells nearby. So nitric oxide leaves the endothelial cell and communicates to immune cells and prevents them from sticking to the vessel wall and moving out of the blood and into the tissue space. It also communicates with platelets in your blood, preventing them from clotting together and blood clots. And both of these are, of course, normal processes to happen at a site of injury. You want immune cells to migrate to fight off infections and clear out damaged tissue. And you want platelets to clot at sites of cuts and scrapes to prevent bleeding. But you don't want this to happen throughout the body. So when you lose nitric oxide in the wrong places, you can actually get these things happening right in the side of the vessel wall. So you get a progression of immune cells migrating into the vessel wall along with some fat deposition, which can build up over time and even rupture, clotting the entire artery. And when this happens in your heart, it's a heart attack. When this happens in the brain, it's a stroke. And so this is how a high salt diet inhibits nitric oxide production, decreases the amount of nitric oxide in these blood vessels, ultimately leading to cardiovascular disease. So this is what we knew going into the project and wanted to understand mechanisms of how nitric oxide is lost with a high salt diet. So are there any questions here before I move on? And if not, that's fine too. Okay, so I'll go ahead and move on then. So like I said, we wanted to understand how nitric oxide is lost with a high salt diet. So I want to talk to you now about an enzyme called Hdac1 or histone deacetylase 1. And I'll be using this sort of green cartoon to represent it in my figures. And this enzyme is one of the regulator enzymes. So it can change how much of other enzymes are being produced, and it can turn other enzymes on and off. So kind of a master regulator of the other enzymes in the cell. And we had studied this one before in the kidney. So kidneys filter your blood, of course, but also maintain a balance of the amount of salt and water in your body. So if you're eating more salt, your kidneys are excreting more salt to bring your body back into balance. And we found previously that this happens because the kidneys increase the amount of Hdac1 in the kidney cells, and this helps them excrete salt and bring the body back into balance. So we wanted to know, is this happening in other cells in the body too? Specifically these endothelial cells, which line your blood vessels, is it an increase in Hdac1 that leads to the decreased nitric oxide production? So we studied this in rats on a high salt diet, and surprisingly, we found that there's no increase in Hdac1 like we thought there might be, but we did see the decrease in nitric oxide production. So, of course, I mentioned these are the doers of the cell, doer molecules, and so maybe they're just working twice as hard and they're twice as active. Maybe there's not more, but they're just doing more. And so we looked at the activity, and that's exactly what we found. We found that these enzymes are almost twice as active in animals on a high salt diet. So now we saw the increase in activity, but what would be really nice if we could fix nitric oxide signaling. So can we block this activity and restore nitric oxide signaling back to healthy levels? So we inhibited the enzyme, and that's exactly what we found. Nitric oxide signaling came back just as if the animal hadn't been on a high salt diet. So now we can build on to our scheme that we're building here, that a high salt diet leads to activation of Hdac1, and this decreases nitric oxide production and leads to cardiovascular disease. So just another break to take any questions before I talk on how we're building on this now.
So Luke, I have a question.
Yeah.
How long do the animals need to be on a high salt diet before you see that increase in Hdac activity?
So we haven't done a time course for this, but people see, so this was 2 weeks, I'll say that, 2 weeks of a high salt diet. And people have shown a loss of nitric oxide signaling as soon as just a couple of days of a high salt diet. And I'm about to show you some data that I think suggests endothelial cells are responding really rapidly on the order of minutes, less than an hour.
Yeah, so that's really interesting. So I'm guessing that what that might mean is Hdac activity could go up and down depending on how a person is changing their salt intake.
Yeah, absolutely. So in people, there's even been studies looking at an hour or so after like a salty soup challenge compared to a low salt soup, and endothelial function changes just within the hour after the meal even. So it definitely seems to be reversible. You can go back on a normal salt diet and restore function.
Great, thanks.
Okay, I have a question too, Luke. So does the increased activity of the Hdac1 affect the quality of that enzyme?
What do you mean the quality of the enzyme?
Well, I guess like the quality of the function, you know, how sometimes things can break down over time, or maybe that's not the best question, but sometimes they don't, work as efficiently, I guess.
Yeah, that's a good question. So I'm going to say this, I'll get to this a little bit in a bit, but we really don't know what it's doing downstream, what it's acting on to change nitric oxide signaling. So it definitely could be that now that's more active, it's regulating other things that it's not supposed to be regulating. But as far as stability of the enzyme, we don't see changes in the amount of it, so I don't think it's degrading faster or anything like that.
Luke, I have a question.
Yes.
If a person is not counting the salt intake, are there any indications in their body or health that they're over salting things, that they're getting too much salt?
Yeah, so
In other words, are there any symptoms?
yeah, if you're in this salt sensitive group, people whose blood pressure changes with the amount of salt you eat, then you would definitely notice an increase in blood pressure potentially. But that's, like I said, not everyone. Maybe with the whole population, it's a little less than half. A really simple one is not always, but often water intake. If you're eating more salt, you tend to be more thirsty and drinking water all the time. That one's maybe a little oversimplified, but an easy one to think about.
All right, thank you.
Another question, Luke, how is it that the increase in salt drives an increase in Hdac activity?
Yeah, so that's the next section. That's
okay.
what we're going on next.
All righty, good.
Yeah, so that's a great segue. So I'll take it there now. So I hope you can appreciate that every time we have an answer, we have a couple more questions. And so the questions we are working on now are, how is high salt activating HDAC1, and how is HDAC1 causing the decrease in nitric oxide signaling? And so I'm going to talk a little bit about this first question that Dr. McMahon just brought up. So we wanted to think about how endothelial cells even know you ate a high salt diet. What are they picking up on? What signals are they reading? So we started thinking about, well, something in the blood is the most obvious. Endothelial cells are, like I said, along this inner layer of your blood vessels. So they're in direct contact with everything in your blood and plasma. So it makes sense to start there. And so we did that experiment. We put plasma from normal salt animals on endothelial cells and saw normal nitric oxide signaling and normal HDAC activity. But when we switched this and did it with plasma from high salt animals, we saw activation of HDAC1 and decrease in nitric oxide signaling, just as if these endothelial cells were from animals on a high salt diet. And this was exciting because, to get to y'all's point earlier, this happened within an hour. So we measured it after the hour, but that means these changes are happening on the order of minutes. So the endothelial cells are really rapidly reversing HDAC activity in response to whatever the stimuli is. So unfortunately, we don't know yet what this stimuli is in the plasma. We have some hypotheses, but nothing concrete. And so we can build a little bit on this model of high salt diet changes something in the plasma, and this activates HDAC1 activity, decreases nitric oxide production, and then ultimately leads to cardiovascular disease. I think this last finding is really exciting to think about clinical applications, because it's not hard to imagine a doctor taking a blood sample and looking for this biomarker and then tracking its progression to give you an idea of how at risk you are for cardiovascular disease progression and give advice on dietary changes or medications that you may need to be on. But even beyond that, because endothelial cells respond so rapidly to this factor, it's a great drug target to change vascular function and health and hopefully prevent the progression of cardiovascular disease and not just track its progression. So with that, of course, I have to thank my mentor, Dr. Jennifer Pollack, and this is a list of lab members, current and past, and then of course our collaborators, especially Dr. Insko and Tony Cook, who have been a huge help on this project, and then the American Heart Association, our funding organizations, and your tax dollars through the NIH, like Dr. McMahon was saying earlier. So yeah, that's the end of my talk. Happy to take any more questions you might have.
Great job, Luke. I'm sure the audience has lots of questions, so please speak up. Don't be shy. This is your opportunity to ask Luke more about his work.
So Luke, I was curious to know, has there been any attempts to associate the levels of increased HDAC activity with either the with the cardiovascular disease, like its actual progression, so that if you're seeing levels at, like, say, that are so elevated, then you know that the patient is probably in an advanced stage of cardiovascular disease already? Or does that make sense?
Yeah. So in the clinic, that's not done. so people have looked at HDACs, not just HDAC1, but the others and other cardiovascular diseases as well. And in general, not always, but in general, more HDAC is a bad thing, and meaning increased cardiovascular disease risk. But hasn't quite made it to the point in the clinic yet to where the assays are kind of a little too complicated to really be assaying directly endothelial cells or just to track progression of disease, which I think is why this factor in the plasma could be really exciting to track these things.
Thank you.
Mm-hmm.
So Luke, I have a few questions for you, and I don't want to, take all the time up. So those of you who have questions, please jump in. But I'm wondering about sex differences. If you've seen a difference between males and females, and if you have, and I see your mentor, Dr. Pollack, nodding her head, so I'm guessing you have seen. Oh no, oh maybe you're just wondering about that. So sex differences. I'm wondering about aging. Is there a difference in the response to high salt diet, with age? And then the last thing is about time of day, circadian rhythms. Have you thought about any of those?
Yeah, so I don't have concrete answers from my data set on a lot of those, but in humans, males and females both have decreased nitric oxide signaling on a high salt diet. Females to a lesser extent than males. And I think, so there was at least one study that failed to find the change in females. And I think it's just because the change is so small, you kind of need a larger sample size in females. And so we haven't investigated the Hdac activity mechanism in our animals, but it could be that there's a difference there through Hdac in females on a high salt diet. The second question with regard to age, so as you age, you tend to have impaired vascular function just as a part of aging, but you also tend to become more salt sensitive. So as you age and your kidneys age, you have a harder time managing those salt loads, and your blood pressure is going to be more likely to rise, which kind of has complications on its own on your heart and other organs. And then time of day, I don't have a good answer for you for time of day. So these were all done in our early morning, which is when the rats were going to go to sleep. And so nitric oxide production is highest kind of during their mid-active period. And so these changes may be different depending on whether you hit kind of the highest point of their nitric oxide activity versus like midday for us, which would be the lowest sensitivity.
Luke, I got a question. At the beginning of the speech, you talked about the South being more prone to high blood pressure. Is that more environmental, or is it just because of our Southern diet?
It's probably a lot of the Southern diet. Unfortunately, there's a lot of nicknames for the Southeast, depending on what disease you're looking at, whether it's the obesity belt or the stroke belt or the stone belt for kidney stones even. And so a lot of that's diet and lifestyle driven in the Southeast.
I have a question. So at the beginning, you talked about the activity of the Hdac1 enzymes and how they like change the production of nitrous oxide or nitric oxide. Okay, NO. And so did you say that at one point you inhibited those enzymes?
Yeah, we inhibited Hdac1 to restore nitric oxide.
So is there like a clinical application to that? Like, could there be a drug or something developed to target those enzymes and inhibit their activity in order to restore the nitric oxide levels back to normal?
Yeah, that's exactly right. So actually the drug we used has been through 4 phase 1 clinical trials to look at dosing and efficacy for lymphomas, so for cancer. And it's in stage 2 clinical trials for other cancers. And so there's promising trials there showing it could be safe for use in humans, and then potentially repurposing those drugs for cardiovascular disease. Yeah.
I have a question. If you exercise more, can you eat more salt?
Yeah, actually, so if you are, if you exercise a lot and are very athletic, then you need to replenish the electrolytes you're losing in sweat, which is kind of why Gatorade was invented. So it does depend how much you're exercising and what your performance level is and the intensity and all of that. But exercise in general is going to improve vascular function and restore nitric oxide signaling. And so these things tend to be additive. It's not like one is the end all be all, but salt along with the amount of fat and sugars you're eating with the exercise and lifestyle kind of all sum together to drive cardiovascular disease.
Okay, thank you.
Mm-hmm.
Can you speculate for us what you think the magic molecule is in the plasma?
Yeah, so the hypotheses we're looking at now are like cytokines involved in inflammation, as well as short chain fatty acids, which are metabolites from the gut microbiome. That's what we're working on now. There's definitely data that a high salt diet changes your gut microbiome and changes the metabolites that they produce. So these microbes that live in your gut ferment the fiber you eat and produce various compounds and funny enough, these compounds really smell kind of like the things that come off of stinky cheeses and really funky fermented things. And those same compounds get in your blood and can be sensed by tissues all throughout your body. And so that's kind of the hypothesis we're looking at now, but don't have anything concrete.
So then those compounds are blocking nitric oxide synthase? Is that what they would do?
So they would inhibit HDAC1 first, activate HDAC1, and then HDAC1, we think maybe through like oxidants and kind of redox proteins, maybe changing nitric oxide signaling.
Okay. Great. Exciting project.
Yeah, thank you.
I have a question about the enzyme in the cells. Whenever it's exposed to a high salt diet, what is the benefit of that enzyme becoming more active? Is it protecting the body from something? And what are the side effects from reducing the activity of the enzymes? Or what have you seen in the rats?
Yeah, so that's
Good question.
Yeah, really good question. The answer is really we don't know. So we kind of only know the bad things it's doing in endothelial cells. So I mentioned in the kidney, it's increased to bring your body back into salt balance. And so if you were to use this as a therapy, that would be one of the really have to watch out for, is preventing your kidneys from getting rid of that excess salt, potentially changing sodium balance in the body and even blood pressure. So you'd have to think about this throughout the body, because this enzyme is expressed in just about every cell in your body as well. It does all sorts of things in endothelial cells, from changing new blood vessel growth into tissues, changing your immune system and how it kind of responds to local environments. And then nitric oxide signaling, like I was saying. So there's a whole host in even cell division, just proliferation of the cells. So it could be doing a lot of things, but why salt drives it up, and if there's some good thing that it's doing also, we don't know.
Great question.
I'm curious, Luke, that was a really good question. So if you guys know the way that HDAC1 responds to other enzymes and different types of chemo signals, have you perchance looked at any of the other signal pathways that upregulate the activity of HDAC1 to see what receptors or how that might be happening, and see if there's a parallel within your salt system?
Can you say that again?
Okay. Sorry if you can't hear me, but here, let me put this right here. So I was just curious. It sounds like we have quite a bit of information about HDAC1 as an enzyme and how it functions. And so I was just curious to know if perhaps you looked at some of the other regulatory pathways that it's active in, if any of those pathways result in an increased activity of HDAC1, have you looked to see if there's something going on there that's similar to the system you're currently studying?
Yeah, so we haven't gone that direction yet. The Hdac activity assay was, that data was just generated even within the last couple months. So we're still kind of very new to us. A lot of implications because it is involved in all of these other things. Yeah, so there's a lot we could look at downstream to see if it's also activated. Not a lot is looked at upstream necessarily at activity level because they're histone deacetylases. So a lot of people who study them are looking at which promoters they're binding to in the genome and deacetylating histones, and not necessarily more active, but where they're going. But yeah, there's definitely some things we can follow up there.
So Luke, you might want to add on that the Hdac in the kidney tubules. So there's an opposite function. So that's actually the kind of good guy.
Yeah. So in the kidney, it's a good guy in that it increases to excrete salt. And so there's this sort of yin-yang effect that Hdac1, it increases in the kidney to help you balance salt and increases in endothelial cells, but to your detriment. Interestingly, it also regulates nitric oxide in the kidney as well, but in the opposite way. And that's a different nitric oxide synthase. But there seems to be this like weird yin-yang between the kidney and blood vessels.
Yeah.
So that makes it hard to be thinking about just giving a patient a pill.
Yes.
Right?
Yes.
And so it would have to be some kind of targeted therapy somehow.
Yeah.
But that's the case with so many drugs that people take, right? It does good things and then it causes side effects, which are the bad things. But I guess in this case, you would just have to find kind of the sweet spot with a dose, right? That would, be split the middle, so to speak.
Yeah. So occasionally stents and such are put in, not for salt, but for vascular damage or thrombosis. And people are coating these stents with various drugs to directly target the vascular.
Yeah.
That could be of use as well.
And when you were talking about the HDAC being beneficial in the kidney and detrimental, I guess, in the endothelial cells, is it a difference that, I think you mentioned in the kidney, the enzyme actually doubles, but in the endothelial cells, it just increases in strength, I think, or you said it's more active.
Yeah.
So is there a difference there in the fact that in the kidney, the enzyme doubles? And
yeah, that's definitely a big difference. I think that points at a big difference at what's upstream, that it's not the same thing turning on Hdac1 in the endothelium that's turning it up in the kidney. And that could be the change in the downstream effects too. So the actual enzyme that produces nitric oxide in the kidney is a different enzyme than produces it in the blood vessels. And so that could be a big point of regulation. Hdac1 changes NOS1, which is in the kidney, in a different way that it regulates NOS3, which is in the blood vessels.
Luke, I think this is a great example that illustrates why we need to know all of the molecules, right? All the steps. You so nicely drew that path with arrows and question marks and dissecting molecule by molecule, and not assuming that it's going to be the same, that the pathway is going to function the same in all cell types. Because you talked a lot about how it's different in the kidney than it is in the endothelial cell. And so it's a great example of why we need to understand and at this level of detail.
Yeah, absolutely. Cells are just bags of chemicals, right?
Yeah. They're very different. Yeah. Any other questions for Luke? This has been a fantastic discussion. So Luke, we hope that you will come back as your story progresses and continue to share with us your discoveries in the making. Got a really exciting story and we're all going to want to hear more.
Yeah, thank you. I'll definitely have to do that. Maybe I can tell you about what that is in the plasma once we feed.
I'm hoping. I can't wait to find out. It's a mystery right now. So we all want to know. So thank you so much. Congratulations on a
Thank
great
you all
talk.
for-
and
Great job,
great
Luke.
project.
Yes, wonderful job, Luke.
So, and thank you all for attending this evening. I know you've all learned many things this evening, and so I would ask that you take one or two things that you've learned and share it with somebody who wasn't attending this evening. That helps us have a broader reach. So I know that you have family members, you have friends, there's someone you can share the new things that you've learned this evening to someone else. And I would like to invite all of you to come back next month to our next session of Discoveries in the Making. Have a nice evening, and thank you again for attending. Good night.
Yeah.
Thank you. Good night.